So to sort of talk about how conservation and translocations fit into into rewilding as a part of it, because they're often kind of interchangeable. The terminology can be a bit interchangeable. So what we're talking about with conservation, so I'm going to start talking about conservation translocations, what they are, the different types, how they work, how they fit into rewilding. And then I'm going to talk a bit about my PhD project and reintroduction kind of as a case study. And then more where I think mathematicians can fit into this and where like we really could do with better methods for modelling and all sorts of areas that could do with assistance. So a conservation translocation is any deliberate movement of an organism from one release site to another. And it is intended to help the conservation, for the conservation benefit of the population, not just the animals that you're moving. So it's a broad term. So it's for the species as opposed to for just the animals being moved. Though it usually is beneficial for those as well. All of these definitions are in the IUCN's guidelines for reintroductions. They're freely available online. So conservation translocations kind of split into two different areas. You've got population restoration and you've got conservation introductions. So rewilding can use all of these really. We've heard a bit about reintroductions, reinforcements, and so these two kind of fit under putting things back where they were originally, whereas conservation introductions are more the functional side of things. So Johan, in his talk at the last one of these, talked quite a lot about restoring functions of ecosystems. So if the species has gone extinct or it's not feasible to put them back, then conservation introductions can be more where we're focused. So a reintroduction is essentially putting a species back where it used to be, so back into its previous range. And one of the conditions on this within the guidelines generally is it's got to be put back after whatever made it go extinct in the first place has been removed, which seems fairly logical, but at the same time, it's not always the case. If it was invasive species, a lot of island systems have had cases where rats invaded. Because of the rats, a certain species went extinct, maybe reptile, maybe bird. And since the rats have been removed, you're able to put the things back. Until the rats have been removed, it's not viable. Reinforcement is where the animal itself hasn't gone extinct, but there's a very low level. It might not be a viable population, and you need to add more to kind of boost the population. So an example could be the European pond turtle. There have been quite a few different reinforcements throughout Europe, Italy, where populations have got very low over time, and just in order to help boost those populations, increase genetic diversity in some cases, that's been beneficial to help those populations. Assisted colonizations are probably the most controversial because this is where you're putting something outside of its native range. Some people think that you'll find a few papers that say we shouldn't ever do these. You will find some people that are very pro doing this. And it's in the idea that sometimes you can't put an animal back into its indigenous range because the habitat's not there anymore. It could be in the future due to climate change, it's no longer climatically suitable. Or the thing that made it go extinct in the first place in that area is still there and we can't get rid of it. Guam is an example of this, island in the Pacific Ocean. Just after the Second World War, it was invaded by brown tree snakes. Brown tree snakes, they've had them there for 70 years now. They cannot get rid of them. They have tried loads of ways. They're quite good at getting them out the army base and not much further. And even when they've invaded tiny islands, that's been a problem. So this bird here is seapeck or Guam kingfisher. It was one of 14 species that was endemic to Guam, only found on Guam. And 11 species, I think, of those 14 went extinct when the brown tree snake was introduced because they had no predators that could get into trees. This one in particular, these birds were put into captivity, and there are about 120 pairs. I might get corrected by my supervisor for that. In the whole of the world, they're all in captivity. So they're extinct in the wild. We can't put them back in Guam. But there's programs trying to put them back into the wild. So that would be an example of an assisted colonization, where trying to put it in where there would have been a kingfisher species, but that species has gone completely extinct in similar areas. So there's a lot of controversy around all of these things. And in terms of rewilding, we could be doing reintroductions, but we also could be doing ecological replacements. This can be where either a species can't be reintroduced because it's extinct, or there's laws that prevent the animal that which was originally there being put back. So in the example of like the Knepp estate that was mentioned earlier, the Knepp estate, they wanted to put wild boar, but because of the Dangerous Animals Act, I'm getting the name that wrong, but they weren't allowed to put boar back. Basically, they couldn't get a license, so they introduced a species of pig that served the same function. It turned over the soil in the same way. So ecological replacements are a species that serves the same function within an ecosystem, but isn't what was there originally. Okay, those are your 4 types of reintroduction of translocation. And the translocation guidelines, kind of very similar to the cycle that was in Rachel's talk and was in the talk earlier, is this kind of thing that when we do a translocation, when we start thinking about it, this is again out of those guidelines, or the UK guidelines, we need to assess what's happening with that species as a whole. What do we know about the species in terms of baseline data? Is it threatened? Is it vulnerable? Has there been a study done on it? Do we have any data at all? Then we need to work out how best to take our conservation action, and potential ways to do that. If it is then made the decision that the best possible case is to translocate, which is risky because translocations, especially early translocations, a lot of them fail. They're very high risk. They often require a huge amount of money and funding, and they're very complex processes, as you will no doubt see by the end of this. If you do decide to translocate, it's then a case of planning and making sure that that plan is as airtight as possible, that you've got something like an exit strategy. So if it all goes wrong, you've got a way, or at least a plan of what you do then. And then once you've implemented it, how you do your post-release monitoring, and most importantly, what is success? And there is a lot of discussion in the literature about what success is. It is not defined. There is no metric that says a translocation is successful if. That is very much up to whoever is doing it. And there is a huge, huge publication bias towards successful reintroductions, which means that we don't necessarily learn everything we want to learn because it's not been published. When things go wrong, we tend to learn more from them. So it's also very, very species level led. So there was a really good review done for all the translocation, all the reintroduction, sorry, I use those interchangeably and I need to not, but for 20 years of reintroduction literature. And in terms of what they reported on, 61% were whether the population had established. So essentially whether it had stayed. 32% on whether it persisted. This can be varying lengths of time. And only 3% of those papers talked about any sort of ecosystem level effects or monitoring of ecosystem level effects. This is not something that historically we're very good at. We are very, very focused on what's happening to the species we've moved, not necessarily how it's interacting with its ecosystem, how it's interacting with the things around it. Now, it could be that those studies don't have the same kind of wow factor and there's a publication. It could be that it's really difficult to monitor these things and too intensive in terms of cost, in terms of time, whether we've got the resources to do it. And there's also publication biases towards, it goes in fashions, so you get publication biases in certain taxonomic groups, for example. So rewilding and translocations. Rewilding is about restoring the function of an ecosystem. And how we can do that in some cases is by putting a species back in. So a lot of it is to do with that. Sometimes it's to do with letting things grow and letting things progress naturally. But in a lot of cases, it is a translocation of some sort. And we generally, our success, if we're thinking about the diagrams from earlier about how rewilding links to restoration and conservation, we're thinking about ecosystem level change, but what the literature is saying is that we're not. So if we want to assess rewilding effectively, we need to start looking on an ecosystem level. And ecosystem level models, some sort of network or ensemble type model, would be really useful, and they are still very in early development. They've been used in other systems, but not so much for reintroductions and thinking about how animals are reintroduced, especially lower levels down the food chain. So what you tend to ideally want with rewilding is if you've got a completely degraded ecosystem, you can start from the bottom and you can restore it or rewild it. Bottom up, you start with the plants, you build up the invertebrate stock, the prey species before you put the predators in. But what you tend to see and what you hear about all the reintroductions that we talked about earlier were big apex predators, top of the food chain. There's a real lack of data on the bottom of the food chain. And that's kind of where we need to be focusing. And with the exception of a couple of these slides, most of them you'll see are very intentionally insects and plants. So because that's really where we need to be focusing right at the moment. There's a focus on all of it, but there is a huge bias in vertebrates and terrestrial land-based systems because I mean, marine systems are a whole nother story because that's all connected. Predators, a lot of predators, which leads to a lot of human-wildlife conflict. There's a lot of mixed attitudes towards lynx, wolves, bears, all of those sorts of things. And it's really difficult to put things in the middle of the food chain. If the predator's been put back in, or if the predator never went extinct in the first place, how do we put a prey species back in and make that population viable without removing the predator, which if your predator's native, you don't want to do. So this was essentially my PhD project. So I was supervised by Rachel, among many others. It was a massive collaborative effort with lots of different charities, well, Mauritian charity, and I was looking at the lesser night gecko. So Mauritius, for those of you that don't know, is in the Indian Ocean at the other side of Madagascar, and I was focusing on this tiny island up here. So Mauritius itself, the main island, is very heavily degraded, like an awful lot of island systems are. It's subject to rats, cats, monkeys, invasive herbivores, goats, rabbits, all sorts over colonial history. And Round Island was lucky that it only had goats and rabbits. So a lot of the endemic fauna weren't eaten. It was just the vegetation that was lost, and that caused the loss of some species, but a lot of them managed to stay. So you end up with this really interesting endemic reptile system, where you've got your top predator, the keel-scaled boa, and also the tail-fed skink. They were found on that one island, Round Island. That was it. That was the only place in the whole planet that those species were. The rest of the way across Mauritius, they'd gone extinct because of invasive species. We found records of them across Mauritius, fossil records, but they couldn't survive rats, essentially. We've also got the Gunther's gecko, which is a predatory species. And then in terms of prey species, there are 4 species of day gecko. There's only one on the island. There are 3 species of night gecko. Again, there's only one on the island. And there's just 1 species of Boja's skink. There might have been multiple species of skink that size. So if we're thinking about reintroducing a night gecko, which is down here where the prey are, I've not included the lower levels because we know pretty much nothing about the invertebrates. There is not a single paper. The first invertebrate survey was early last year, the first proper structured one, and this island's been worked on since the 70s. So it's a relatively long-running conservation project. There's been a lot of planting and reforestation. It's been under different management a lot of the times. Durrell have been heavily involved, ZSL have been involved at various points, Mauritian government. So it's a big collaborative effort, and that often means that things change quite regularly. But there is a 100-year plan for the restoration of Round Island, and it is fed into the government on kind of a 5-year basis of this is what we want to achieve. So lesser night gecko was in our 5-year plan for this section, and we wanted to put it in because the night gecko on Round Island was quite a lot bigger than the night gecko that we found on some other islands, and it fitted into a slightly different ecological niche in terms of what its predators would be. So we wanted to put a prey species back, and obviously that's not easy when you've got species that are vulnerable. All of those are vulnerable. Boja's skink's critically endangered, and both of those are vulnerable as well. So we can't really do too much to disturb this ecosystem because it is so vulnerable already. So when we assess the conservation situation of the species, the lesser night gecko itself is vulnerable, It's found in Mauritius. It's found on 4 tiny islands. 2 of those are less than 0.1 kilometre square, so small rocks in the sea that you can walk from one side or the other to in like 5 minutes, which will probably go underwater when sea level rises. So it's in a vulnerable state, and the islands that it was on, where the biggest population was quite close to the mainland, often trespassed. The chance of a re-invasion of invasive species is higher than it would be on Round Island, which is a nature reserve patrolled by wardens all year round. So that was kind of the best place for them to be. We could do it. So we thought, in terms of conserving the species, there's no better island than Round Island to put them on. And we looked at our options and we decided that a translocation would be the best case scenario. So in terms of planning, what we wanted to do was estimate the survival of one of the other populations that had been translocated in 2011. It had been translocated to a predator-free island, so it was a slightly different system, but it was still a good model of how the population grows when you put them somewhere new. I also wanted to do a literary review of lizard translocations. In reality, we tried using standard capture-recapture models because that was exactly what the data had been collected to use. So it was capture-recapture survey. They have a unique pattern, so they can be identified individually. We were using Jolly Seaver models because we wanted an estimate of population size. And we were coming out with ridiculously high uncertainty because some of the surveys over a 3-day period, they would find 5 animals. So your sample sizes are tiny. And in terms of literature review, there were about 3 papers published on small reptiles being translocated. And that is potentially because quite a lot of them fail and then they don't get published. Or a lot of them are done by consultants and end up in grey literature. So there was an issue there. So how do we plan translocation when we have got very limited model approaches, very limited demographic information, and very little literature? Well, we, with a lot of help from Rachel, modified a standard Jolly Seaver model. I can't talk about the specifics of that. The QR code takes you to the paper. It is now published. Essentially, we created a bespoke Jolly Seaver model that accounted for the fact that some animals have been translocated, and that allowed us to reduce our uncertainty. We did a simulation study to compare that model to a standard model and found that it did reduce the error, especially when the population was very small and very hard to find. And the other parts of that model were constraining the kind of entry probability because you've got a biological period where they can't be breeding because it's not feasibly possible for there to be new entrants to the adult population. And then literature review wise, we ended up working with expert elicitation. So we did a semi-structured interview at the World Congress for Herpetology and spoke to 25 experts, a lot of whom do some consultancy work, and a lot of their experience was through consultancy as opposed to projects, or they were projects that were so recent that they hadn't been published anywhere. So they had a really good idea of what sort of release strategies we should be considering. And they recommended that we built fenced enclosures to allow the population to build up a bit before the predators were allowed anywhere near the lesser night geckos. So they also helped us identify potential risks, and the number one risk was dispersal. There is a trend in lizards that if they are stressed and you put them down somewhere, they run in all directions. Now, lesser night geckos are about this big, including their tail, and once they settle in a spot, they stay within the same 3 meters squared almost their entire life. So if they run in opposite directions and settle, we're going to end up with 30 lesser night geckos on an island that are not breeding, which doesn't help even in persistence. So that was obviously, we needed to make sure that they were anchored to a site and try and keep them close. And there's one study on geckos that says that putting them in a fenced area does reduce dispersal when you take the fences off. Then we had to find a site that was accessible, and this is what some of Round Island looks like. If you look at the horizon, you can see that's straight. This is the island. It's all on a gradient like this, so it's all really steep. It's all really rugged. Trying to build a fence on this is not the easiest thing in the world. So I spent a good few weeks wandering around this island looking for somewhere flat enough to fence. And also we wanted a nice rocky outcrop. They like this sort of nice kind of crumbly rocks with lots of holes in them. One of my PhD supervisors, in fact, during his PhD, had looked at crevice size in particular and measuring how big a gap they liked. So I had that data, which was good. Then there were the other night gecko species, which I mentioned, the Durrell's night gecko. They eat similar prey, but because that's insect prey, they have kind of they'll eat a larger version of what the lesser night geckos would eat, which means that if the Durrell's night gecko were there probably should be a food source for the lesser night gecko. And as we had no data on invertebrates, that was the best proxy we had for if there was food availability. It was if on this bit of the island there's Durrell's night gecko, then lesser night geckos should be able to survive. There are giant tortoises on the island. They were actually an example of a I can't even remember the name. Sorry, I've gone completely blank. They were put in as a replacement because the Mauritian tortoise went extinct due to people eating it. And that was, so this is a species from the Seychelles, the Aldabra giant tortoise, and that was put back in because tortoises were really important seed dispersers on the island. They would eat fruits from the palms and disperse them all the way across the island. So this is quite a good example of restoring the function of an ecosystem with a non-native species. And also because they were so large, if something didn't work when they were put back, they were quite easy to find and take off again in that case. So it was a very clear exit strategy. But unfortunately, tortoises are also bulldozers. And if they see something they want to eat, they walk straight towards it. So we had to plan our fences so that they weren't on standard tortoise routes. And they have quite standard tracks. They've got their areas they like. So we avoided those areas of the island. We did get the odd bit of tortoise disturbance, but it wasn't too bad. Predators. We wanted to avoid especially juvenile predators. So this is a juvenile Gunthus gecko. It's about this big, and that would quite happily eat a lesser night gecko. We had a little bit of data on the most dense areas for the different predator species on the island, what sort of habitat preference they had, and where juveniles were found most commonly. So we avoided those areas as much as we could. It also rains a lot in the wet season, so we needed to find somewhere that wasn't a great big water run so that our fences got washed away or the habitat didn't get washed away. So combining those things, these are all things that I suppose are extra complications on top of biologically they're a prey species in a system full of predators. So we did build fences. This is one of the fences, and we had 30 geckos per enclosure. We had 4 of these enclosures on the island, all quite close together so that eventually the populations should join together. We put this predator-proof fencing, highly sophisticated black plastic, which things with sticky toes aren't very good at climbing shiny surfaces, so it stopped quite a lot of the geckos and the skinks. We also greased the top of it to stop the geckos getting in or out, and it was buried underneath to stop anything digging under. And we also put some artificial refugia in, which are just like extra habitat for them to help them join the best bits of habitat together. So that was the release. So we designed and planned our translocation, and we helicoptered them over from the neighbouring island, which looks very sophisticated, but almost didn't happen at the last minute because government were another partner on the project, and government changed their mind about helicopters within a few hours quite often. But it did happen, and we did get them across. The reason for a helicopter was that there's a lot of invasive plant species and invasive species on the island that they are resident on, so we wanted to get away them to be transported without any opportunity for more hazardous material to get into the boxes. And when we got them, we had to check the outside of every single bag for seeds, for insects, just to make sure that nothing that was on the other island that wasn't on Round Island got there. So this was kind of our biosecurity measure. Right, and then monitoring our outcomes. Post-release monitoring, this is what the back pattern looks like. This is kind of their unique ID. You can use photo ID software on it. You can do it by eye, but it takes quite a long time. So that's what we did to develop our capture histories for our capture recapture. More things that occasionally go wrong in the field. My I was meant to have an assistant from the start. Her visa didn't come through for 2 to 3 months, so I didn't have an assistant, and that meant she had to be trained by someone else. There was no overlap between us being there, which meant that the way she was doing it was correct, but I wasn't there to train her or like meet her in person. And that also meant that her first few surveys, one was with someone that helped her, her second one was on her own, and the capture probability really drops, which is not her fault. It's a learning process, but that learning process should have happened while I was still there. So that kind of affected our probability of capture. Then there was a cyclone in between 2 of her surveys, which didn't completely break the fences, but it wasn't ideal. And then all of this got delayed because of COVID. It was 2 years late, so it came into the 3rd year of my PhD, not the end of the 1st year, which meant I had 6 months worth of data to analyse to get a chapter out, which we can't really tell much from 6 months, especially in a cryptic species that are really difficult to find. So these are all the constraints of a perfectly ordinary translocation. If you're working with a small cryptic species, none of this is unreasonable to happen. And that's kind of where we need to think about how we're analysing the data at the end, so that we can work backwards and create a process that is suitable for a good translocation. So this is kind of where I think, going back to this cycle, where we can really get some benefit from working more with mathematicians and statisticians and social scientists as well, is the kind of the baseline assessment. When we're getting our baseline of what is the status of these species, we really could do with methods for small data. Big data has been a fashionable thing for quite a while now. Everyone likes big data. In conservation, we don't often have big data because we have got a small amount of animals, and that it can be collected, but it's time intensive. Conservation, we're often financially limited as well. So if we're looking at a system where we are limited on money, doing really intensive surveys isn't necessarily feasible. So when we look at we really could do with some better methods for analysing small data and on an ecosystem level, because changing one thing in an ecosystem can have a huge effect on the rest of the ecosystem. It's all connected. In terms of evaluating options to achieve our goals, so when we compared release strategies, ideally we wanted to model what was likely to happen if we used different release strategies. We didn't have enough information to be able to do that all the time, to develop the methods to do that. Again, sparse data. We had expert elicitation data, but we ideally we would have liked to have done something quantitative with that as opposed to just qualitative. And again, ecosystem level models to look at what's going to happen to not only the predator, but the prey species. And going back to the initial kind of conservation status of the species, if we're working on a species and we don't know much about its food source, can we collect data before we do a translocation to look at that food source? Is that resource there? And can we use a proxy? With designing translocations, clearly defining what is success. What are you aiming for? Our aim was for within 6 months our species to be established, because we knew that over 6 months we wouldn't necessarily have me working on the project and it was handed over to the local charity. So our short term was that they were persisting within 6 months, which they were, but at the same time it wasn't fully successful because we need long-term data to know if it was actually a success. So our short-term aim was met, but our long-term aims were more difficult to define. So also predictive models, what's likely to happen under different strategies, optimal numbers of individuals for translocations. The metric of 30 was about on the density of lesser night gecko that are found in the wild, and the most that you could fit in a 10 by 10 meter enclosure without any kind of negative density effects happening. But again, it would have been nice to have done something predictive with that. And having a good exit strategy, having an exit strategy that will work, or a very defined point where you know that this population is dropping at a point where we might need to move those animals out again if you're working with very threatened species. And monitoring. we, as conservationists that are limited by funding, ideally we want to know the minimum we need to do in order to calculate whether we're successful and to monitor the species so that we can get an estimate of survival. Feasibly, it's really important to talk to both the statisticians and the conservation side in order to work out if that monitoring could be done and if the data from it is actually useful. And yeah, the metrics that would trigger an exit strategy. So most of all, we want methods for small data because small data is what we've got, and that's really difficult. And whether it's splitting models down into more bespoke stages, that could be an approach, but it's difficult. And as sort of a statistician myself, not fully a statistician, more of an ecologist, I don't know how you would go about improving that massively. Accounting for like uncertainty, there's always going to be uncertainty. If we can minimize that, that would be ideal. If we can minimize the risk, that would be great. But yeah, that would be a nice way to elaborate on that. Make it accessible. Not all conservation managers will have a good understanding of how R works, how coding works. Can we build some sort of front end for it in our Shiny app where we can put the numbers in and get good, reliable estimates out? It's kind of when you're building these tools of identifying who your target audience is, making sure that you're actually creating something that's appropriate for the audience that doesn't just sit somewhere and is never used. If you want to play with model systems, island systems are really good because you can treat them as a closed system, or fragmented habitat. If you've got a habitat fragment, that's quite a nice way to think about modeling an ecosystem. And it limits the number of species if you've got a limited range. And ecosystem level models. So there's a bit of development on ecosystem ensemble models, which are looking promising, but and yes, success metrics. Cryptic species are always difficult. Cryptic, I mean anything that is difficult to find, small, well camouflaged, has large periods of its life where it's in the deep ocean, that kind of thing. And marine ecosystems, I know that there's been quite a lot of work on marine within various of the sort of biostatistics groups across Scotland particularly, and it's good, but marine reintroductions, those are barely starting. How on earth do we do that together? I think if you want a real challenge, looking at how on earth you introduce something into a system that is that big is fascinating and maybe doesn't have an answer. But sorry, that was done very quickly. So thank you. I'm happy to take any questions.